So, team name, genetic goals, problem statement ID SIH25042, problem statement title, identifying taxonomy and assessing biodiversity from environmental DNA datasets, theme miscellaneous, PS category software, team name, genetic goals. So welcome to our interactive dashboard. The interface is designed to be simple yet powerful so that scientists and policymakers can use it without technical barriers. From here, user can directly upload their DNA data and explore biodiversity insights. So basically scientists will take the reference or collect the data from the segment water DNA data and convert them into the FASTA files, and we will take the input of that FASTA file. Using generative AI, DNA birds and embeddeds and analomy detections, we not only classify known species but also detect the potential novel ones. So basically you can see here I will upload the database of the species and it will be start quality controlling analyzing. This way they will be classifying mode, not only telling us the when species are present but also give us a meaningful ecological insight about where they live and how they functions in the environment. So you can see here total reads are 45,000, quality passed are 42,000, its average length, its GC content, its duplications and overall quality process. So now we will move on the classification sections. So if we start analyzing it, classification section, once sequence are processed, the platform classified them against multiple reference datasets. So you can see here exact match species, highly similar species, medium similar species and novel species. So this is Thalas Ceosacerea oceanea. So this species is exactly matched with this particular taxonomy with the 98.7% similarity, having the database from BLAST. So its characteristics match are kingdom is belongs from the Eukaryotea, phylum is Bacillophytia, its class, order, family, genus, species. And now ecological information, habitat, marines, planktonic, depth range means 0 to 200 meters, means this particular species lies at this particular depth range and its abundance is average about 1247 and its characteristics. So next species is highly matched with this particular taxonomic details, so with 89.3% similarity, its kingdom, its phylum, class, order, family. So you can see here its ecological information, also its depth range is surface to 100 meters. So next is medium levels, Rogostoma species. So particular species is matched with the taxonomic details of Rogostoma species with the 73.2% similarities. Its kingdom, phylum, class, orders, ecological information like depth range, its depth range is about 100 to 4000 meters and its abundance is 234. In this way this will going to show you. This way the classification model not only tells you what species where are present but also gives you a meaningful ecological insights about where they are living, how they are functioning the words, what is their depth levels, what are their characteristics, what is their abundances, everything. So now after classification we will move on the ecological section. Now we move into the ecological insights where raw DNA sequence are turns into a clear pictures of biodiversity. So you can see here from our database there are 12,450 sequences matched with Bacilla rheophyta, about 42.3%. So what is this? Basically plankton that can both photosynthesis and feeds on other organisms, and 8,920 sequences match 30.4% with Dinoflagellata. What is Dinoflagellata? Basically it is a planktons that can both photosynthesis also. So next is Seracosa. From our database there are 4,230 sequences match 7.1% with its. So what is this? Basically it's small amebia like microbes that recycles nutrients. Next is Ciliophora. From our database there are 1,650 sequences match 5.6% with these characteristics. And next is unknown, means sequence that don't yet match any reference database, hinting at undiscovered diversity. So to support this biodiversity matrix like the Shannon index, Simple's index and species richness give a statistical view of diversity and domains. I thought these sections connects the dot between the genetic data and ecological meanings and then providing both quick snapshots of community health and tools to explore species interactions and details. So now we will move on the novel detections. So basically it will be going to analyzing for 3 to 4 seconds. After analyzing it will detect the novel species. So you can see here 4 novel clusters are detected, 123 sequences are there, 2 highly novel characteristics are there and 86.8% average similarity. Basically an average similarity of 86.8% showing this sequence are quite different from the known references. So this is the novel cluster. So you can see here there are 43 sections: alpha section, beta section and gamma section. 47 sequences are matched with the alpha section, that they are 94.7% novels and different from the other species about 87.3%. So their characteristics, some type it will be matched to the this taxonomical match. So Kinetoplastida sp, its kingdom basically Eukaryotea, similarity is 68.2%. Also you can see here depth range, locations, Abyssinian plants, at what temperature they are living or they are surviving at 2.1 degrees Celsius, what are their habitats, and this is the representation sequence in the form of adenine, thymine, guanine, cytosine. So next is our species beta. 23 sequences are matched with the beta species and they are 89.3% novels. Also you can see here its ecological content, sequence and taxonomical match, and 34 sequences from our input database are gamma database having a 97.2% novel species. Their depth range, location, temperature, habitats and representation sequence. Basically, and we will also providing here confidence level, that how confidential our software about this information: medium level confidence, low level confidence and high level confidence. So now we will move on the next section, which is geographical maps. So basically you can see here this is all samples. This is the depth level. If we change the depth level, it will be changed according to its. This is the highly novel, greater than 90%, medium low level novels about 70 to 90%, and low is less than 70%. So these sections place biodiversity finding into their geographical and environmental context, helping us to understand where species are found and how they are distributed across the different habitats. So at the selected species, the Pacific abyssal plain samples were collected are around 3200 meter depth, where the temperature is cold about 2.1 degrees Celsius. A research depth is 34 taxa with diatoms. As the diatoms groups, the site shows about 78.5% novel characteristics and total abundance is 12,450. This is the 3D representations and about the depth levels and how much characteristics and organisms are present at the particular depth level. So our next section is report and export. Means if a scientist want this data, researchers wants this data, they can download this data from here. So all history will be stored here. Scientists can able to download whole the reports. This is the raw data. Means if they wants to download the raw data, they can also able to download the raw data. This is the visualization format. If they want to download in the form of visualization graphs, we will also provide these features. So you can see here supported extended formats, compatible formats for download streamlines. Bioinformations, QMAI to OTU tables, biomes, sequence data is FASTA, phylogenetic trees, NWK, data analysis, how data analysis works, how visualization is going to works, and how data citation and usage. And these are our data usage guidelines. So this is our overview of our prototypes. And this is just a beginning. So we are now working to integrate a global biodiversity database like GenBanks and bolts to increase the accuracy or problem statement than others. By combining this global knowledge with advanced technologies such as generative AI, reinforcement learning, graphical neural networks, our system will evolve around the world. Powerful tool for real-time biodiversity monitoring and worldwide. Problem statement is that we are losing a species faster than we can discover them. So current DNA testing methods are too slow, too costly and too complex. Data is scattered, results often come late, and by then it's impossible to save what's already lost. On the top of that, only a few experts can work on biodiversity research, while ordinary peoples and smaller labs are left out. So basically our solution for this problem statement is that our platform makes biodiversity study faster, smarter and open to everyone. With AI, we can spot species early. The cloud makes it scalable and affordable, and a blockchain ensures data is trustable and secure. So researchers, conservation teams and even citizen scientists can now work together in real time to protect nature before it's too late. So first problem for this is hidden biodiversity knowledge gaps. So many species remains undiscovered due to incomplete genetic references. So impact is that loss of valuable biodiversity insights and missed opportunities in conservation and medicine. So our solution is that AI DNA birds for hidden genetic patterns, generative AI for novel species discovery, unified biodiversity knowledge gaps. So tech stack for this problem is that Python, having the frameworks like PyTorch, TensorFlow, Neo4j GraphDB, PostgreSQL and fast APIs. So next problem is that inaccurate classification and fragmented analysis. Traditional method misclassify species and fail to merge multi-ecosystems data. So impact is that wrong biodiversity reports and poor ecosystem policy. Our solution is multimodal AI for soil, ocean, river, environmental DNA, automated pipelines for real-time classifications, unified ecosystems levels mapping. So tech stack for this problem is that Nextflow, scikit-learn, Spark, Ray and MongoDB. So next problem is that delay conservation response, slow data processing, delay threat detection. And impact is that late conservation action leads to species and habit losses. So our solution is that AI DSS for real-time alerts, predictive analytics for early risk detections, interactive decision dashboard. So tech stack for this problem is that React.js Plotly. Basically, Plotly is used to represent our solution in the form of graphical representation. Fast APIs, AWS, Lambda and Kubernetes. So next problem is that lack of trust in shared data. So researchers' data is scattered, unverified and non-transparent. So impact for this is low collaborations and unreliable results. Our solution is that blockchain for tamper-proof sharing, smart contracts for secure access, transparent audit trails for research. So tech stack is that Ethereum, IFS, Fabrics, Node.js and Express.js. So next problem is that high cost and low accessibility. Problem: environmental DNA analysis is very expensive, resource heavy and complex for citizen scientists. So impact is that limited access and slow adoption of biodiversity monitoring. So our solution for this problem is cloud-based scalable AI pipelines, federative learnings for global collaborations, simple portals for researchers and citizens. So tech stack for this problem is that AWS, Google Cloud Platforms, Kubernetes, Docker, Streamlets and Tailwind CSS. So what are the uniqueness we are going to provide for our solution for this problem statement? So uniqueness are: first is 3D Ocean Explorer, means explorer oceans and biodiversity on a 3D interactive globes with a depth layer. Next is hidden species discovery. AI finds new species even those are missing from existing database. Confidence score: every result shows how reliable it is the systems with AI backed scoring. So next is federated learning collaborations. Labs worldwide can work together without sharing a sensitive DNA data. Biodiversity knowledge graphs, connected DNA, taxonomy and locations into one smart searchable networks. These are the uniqueness. So what is our flow of the solutions, how our prototype is going to work? So I am going to explain this. So first it will be starts. Admin create the project, assign unique code, allow users to join project. These three steps are happening in the backend part. So next is researchers upload the sample in the form of the FASTA file, which represents our characteristics of the biodiversity. So next is system automatically sorts and group up the data. Data processings. If a FASTA file is matched to our existing data, then it will be linked to a species and approve the submissions. If it will be not matched, we will detect it as a novel species, so mark as a new candidate. So experts will check that this species is really novel or not. If they will be approved, this will be accepted and this will be indicated as a novel species. If they will be rejected, then against research will be happen on this particular species. This is our flow. So next is our biodiversity integration systems. Value propositions, key resources, key activities, custom segments, revenue streams, customer relationships, channels and key partners. These all things we are going to implement. So we are building a scalable and futures ready platform that can support scientists, policymakers, conservation researchers, solving global ecological challenges. It also helps to solve the problems on the natures and biodiversity. So these are problem statements and solutions. Thank you so much.